Welcome guys, today's topic is about your Ames test, very simplest but the most commonly used test to know whether a substance is a mutagen or a carcinogen. So before going to this thing, let me clarify you what is the basic difference between mutagens and carcinogen. Well, you can see here the carcinogens are those agents which lead to the cancer that converts normal cells to cancer cell. So this is the thing, you can read it out, but the basic thing about this is mutagens belongs to the carcinogen. Mutagens affects the cancer, cause the cancer by creating mutations in DNA. But carcinogen is a broad sense which includes these mutagens. It also deals with your cancer which is related to your epigenetic regulated conditions, for example like your CpG isolate methylated conditions, organ interference, expression of a gene by the enhancer promoter, histone mediated methylated histoproteins. So in this way the expression of a protein is a change and that leads to the cancer. So this is a very bright difference between the carcinogen and the mutagens. So the mutagens cause a mutation, belong to the carcinogens. Carcinogen cause cancer, but it cause cancer in two ways. That is by mutation, changing the DNA, or without changing the DNA. Without changing DNA, it helps. Takes the help of your epigenetic regulators. Okay, so this is the basic thing. So let's move on to the test. You can see here Salmonella typhi is that mutant bacteria that is taken for this experiment. As you can see here in this this mutant is because it is unable to produce amino acid, or we call the histidine. The histidine is your amino acid which is unable to form by the Salmonella typhi of a mutant type. So whenever this mutant type bacteria is put inside a homogenized liver cells of rat and allowed to be metabolically active test compound, for example, you can see here this is a test compound. That means this compound is to be checked whether this compound is a mutagenic or carcinogenic property. So for that we have just taken the bacterial cells that we put inside that we mix in with the help of those test compound and homogenize rat liver cells and allow for incubations in the culture media. So two things will be the result. Two results could be possibly occur. One, there will be no growth of the bacterial colonies. There will be growth of bacterial colonies. So in those cases where there's a development of a bacterial colony occurs or found, that means there is some kind of mutation, a kind of point mutation must be there that alters this mutant histidine molecule production, histidine amino acid productions that lead to a mutation that creates this positive, that means a bacteria with able to produce histidine metabolic capability. So those bacteria which undergoes mutation, they will be able to colonize on the plate, and this is possible because of this compound which is a mutagenic compound. If this is not a mutagenic compound, then it will not be able to alter or changes, cause any mutation. And as there is no mutations, so this bacteria will not be able to proliferate because they can't use the histidine amino acids to form their protein to becomes to able to proliferate. So this is the thing, that in this way this test is done to check whether it is a tumorigenic or carcinogenic compound. In this picture you can see increase in carcinogenicity And in this increase in mutagenicity is almost in very close approximately, and the most mutagenic property is found in your aflatoxins, even in very low concentration of about 10 to the minus 3 milligram per kg per day will also cause a tumor induction. Similarly, in this case, you can see the mutagenicity property of this aflatoxin is also higher in the very low dose, about a microgram, to induce the hormonal revirence, that means the mutants opposite. Thank you very much. I hope you understand. Not able to understand, comment below. Thank you.